Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending. I'm your moderator, Ellen Capon, Editorial Assistant at Drug Target Review. Today's speakers will be Vanessa Porcalab and Maud Sigalo. Following their presentation, we will move on to a live question and answer session where you can pose questions to today's speakers. Please remember you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So without hesitation, may I pass over to Vanessa and Maud.
Thank you for your introduction. Hi everyone, it's a pleasure to be here today. We will address in this webinar today several topics related to kinase drug discovery programs. Before starting, I would like to replace the standard drug discovery workflow and the role of biophysics at Eurofins Discovery. Biophysics are commonly used in hit finding, hit to lead, and lead optimization phases. We offer custom services for small molecules from standalone projects to integrated drug discovery programs. So today we are going to present you the biophysics strategy that was deployed for a specific kinase drug discovery program. Before starting, we will do a quick introduction about us. My name is Vanessa Porcalab. I'm a head of biophysics at Eurofins Discovery CREP. I have a background in biophysics, working from fragment to antibodies characterizations. For several years now, with my biotech and CRO experience, I'm driving biophysics projects and leading a team of scientists involved in drug discovery programs. It's a pleasure to be here today with Maud, so I will let Maud introduce herself.
Thank you, Vanessa. I'm very happy to be speaking with you today. My name is Maud Sigalo. I joined Eurofins Discovery as biophysics project leader in February 2023. I work closely with the clients to define the most effective biophysical assay development strategy for their project. I have a PhD specializing in the perception of sweet and umami tests mediated by GPCRs. And then I did a postdoc in biochemical characterization of nanobody bindings to the cystic fibrosis transmembrane regulator for therapeutic purpose. Before joining Eurofins Discovery, I was project leader in biophysics at a biotech company working on oncological drug discovery. And now I'll let Vanessa introduce the importance of biophysics in drug discovery.
Thanks, Maud. First, I would like to define with you what are the role and the scope of biophysics. It will be a quick reminder for some of you or a quick introduction for the others. Biophysics techniques is commonly used in drug discovery from compound profiling to mechanism of action. Working mainly with recombinant proteins, the biophysics allow us to characterize hits. Our goal is to understand the relationship between kinetics, mode of action, and molecular interactions. Thanks to complementary biophysics approaches, we can look on the strength of the binding, the kinetic, the thermodynamic parameters, the stoichiometry, and finally, we have also insight regarding the recombinant protein quality. Based on the request and the aim of the project, those information as KD, K on, K off, melting temperature bring valuable data for compound ranking and selection. As example, typical projects that we are offering in this hit finding stage, we can perform fragment screen campaign with a defined set of fragments that will be a starting point for fragment growing, or we could also introduce biophysics as an orthogonal assay after a HTS campaign. After validation of hits using functional assays, we are confirming the hits with their binding properties for the target. Regarding hit lead and lead optimization, we are providing support for medchem analysis and insight into the mechanism of action. We know that kinetic can help to predict in vivo pharmacology. If we have a specific request, as you would like to know the hit selectivity for the target, the binding stoichiometry, or if you are interested to understand if your compound is allosteric or orthosteric binders, biophysically can help you. Typically for kinase drug discovery program, we would like to see if our hits bind the ATP pocket or if it competes with other well-known inhibitors like inhibitors type 1, 2, 3, etc. All of those projects, including screening and hit characterization, are possible thanks to our cutting-edge equipment. Here at Eurofins Discovery, we are working with the most recent biophysical technologies. From the left to the right, you could find the BioCore 8K+, the Dianthus for the MST trick technologies that include also the spectral shift approach. We have also the PIC ITC and the BioRad devices. All of those technologies are complementary and have some advantages and limitations. The recombinant protein, here in our case, the kinase, can be mobilized on the chip for SPR, can be fluorescently labeled for trick spectral shift experiment. It could be also in solution for ITC experiment. And finally, for thermal shift assay, we incorporate dye to monitor the unfolding process of the kinase. For screening and determination of KD value, BioCore 8K+ and the MST trick technologies fit perfectly for the need with their 384 well-plate format for compound testing. Additionally, we could extract several types of information based on the technologies as binding constant, kinetics, thermodynamic parameters, stoichiometry, and melting temperature. For routine quality control, we recently acquired the Pentaprometheus for protein QC, incorporating the nano-DSF and DLS technologies, and we have also the nephelometry assays for compound solubility. So let's start with a specific case study that was developed here at Eurofins Discovery. Several drug discovery programs are ongoing, and the aim here is to introduce two of them. The current PIM3 fragment screening campaign using the spectral shift technology and the PIM3 degraders. So why we select PIM3 as a target for hit finding? The PIM3 kinase is a very attractive target for cancer therapy, as it was described as promoting immune evasion. The kinase belongs to the serine-threonine kinase family and involves three highly homologous enzymes, including PIM1, PIM2, and PIM3. They have similar function and kinase domain, but differ in their distribution in tissues. The aim of PIM3 drug discovery programs is to find PIM3 inhibitors and/or degraders using several approaches. The first one is the HTS campaign, with biophysics studies used as orthogonal assay. Promising hits were identified in terms of activity, binding, and selectivity for PIM3. And if you are interested in this strategy, you can take a look on this webinar available online. The second approach is the fragment screen campaign, topic that will be discussed today with you, and finally, the PROTAC strategy. I will give the floor now to Maud for presenting the scientific data that was generated for fragment screening.
Thank you, Vanessa. The quality of recombinant protein is key for the development of robust biophysical assays. For this study, we used the recombinant PIM3 produced in-house by Eurofins Discoverex. First, we checked the protein thermal stability in the storage buffer and the effect of the fluorescent labeling using nano-DSF. The Prometheus Penta enabled us to work at a low concentration, 50 nanomolar, which is the concentration obtained after labeling. This wouldn't have been possible using the traditional thermal shift assay with Cypro range. As you can see on the bottom right, the first derivative of the fluorescence at 350 nanometers indicate a melting temperature of 33 degrees in the storage buffer. This is the green curve, and 31 degrees after histag labeling, the purple curve. These results show that PIM3 labeling combined to the buffer exchange has a slight destabilizing effect, but does not promote PIM3 unfolding. Before going into the details of the feasibility study, I would like to draw your attention to the screening cascade we followed. As I said, PIM3 was produced in-house, and we used a subset of our library of fragments for this study. We perform a feasibility study to select the best biophysical approach for the screening of fragments. Then we did the single-dose screening, enabling a first hit triage. The hits were confirmed by dose-response experiments, allowing the ranking of the fragment by affinity. And next, we used TSA as an orthogonal assay to identify PIM3 stabilizer. And finally, a competition assay was set up to obtain more information on the mechanism of action for this fragment. An activity assay and structural biology approach complete the biophysical cascade. Let's move to the feasibility study we performed to select the best approach for the fragment-based screening campaign. We compared MST spectral shift technology that requires fluorescent labeling of PIM3 with SPR requiring protein immobilization. ATP gamma S, a non-hydrolyzable ATP analog, was used as a reference compound to validate the assay development. During the SPR assay, two immobilization strategies were tested, enabling either oriented immobilization or random immobilization by amine coupling. Fortunately, none of these strategies allowed the detection of ATP gamma S binding. The signal variability observed was induced by structural changes of PIM3 over the surface. In parallel, we developed the MST spectral shift assay. We obtained a good labeling level as shown on this graph. The initial fluorescence was around 5,000 counts and was not affected by ATP binding. We obtained a dose-response effect of ATP gamma S in the micromolar range using the spectral shift readout, but not the MST trick. Based on this result, we selected spectral shift technology as the primary assay for the fragment-based screening campaign. PIM3 fragment-based screening was performed with a subset of 826 fragments from a library of 3,133 fragments using the MST trick and spectral shift approach. Fragments were tested at the final concentration of 600 micromolar in duplicates. Hits were defined as compound, inducing a variation of the response higher than 3 times the standard deviation of the DMSO control, represented by the blue arrow in the 1D screening panel. Hits are shown in light blue circles. It is important to note that in spectral shift, hits can positively or negatively affect the response, corresponding to a blue shift or a red shift of the emission wavelength. Our positive control, ATP gamma S, was regularly monitored to assess PIM3 stability and is depicted as a triangle, as orange triangles. We obtained a hit rate of 11%. The fragment binding was validated by DRC. And before moving to the results of the DRC confirmation, I would like to highlight that the compound dispensing was done by our compound management team by eco-dispensing. This workflow speeds up the screen and limits the compound consumption and the manual pipetting. Dose response titration were then performed on 87 fragments in duplicates. You can see, for example, on the top panel, the dose response curve of the best PIM3 binder. this fragment shows an affinity of 23 micromolar, a good signal-to-noise ratio of 13.5 with a saturation of 95%. In the middle of the screen, you can see the frequency distribution of PIM3 confirmed binders ranked by affinity. More than 79% of hits were confirmed with the DRC and could be ranked by KD. Thermal shift assay, or TSA, was used as an orthogonal assay to assess the effect of fragments on PIM3 thermal stability. TSA is a simple technique where the fluorescence of protein solution is measured in a temperature gradient. We use the Cypro orange dye, which binds to the hydrophobic residue that get exposed as PIM3 unfolds. The first derivative of the melting curve enables the determination of the melting temperature. We use the pan-PIM inhibitor named AZD1208 to set up the experiment. As you can see on the graph in the middle of the slide, AZD1208 strongly stabilized PIM3 by more than 18 degrees. The blue curve corresponds to the denaturation of PIM3 in absence of compound, and the green curve in presence of AZD1208. Then we tested a selection of 33 fragments in triplicates at 600 micromolar final concentration. As you can see on the histogram, PIM3 binder exhibit different effects on PIM3 thermal stability. They can be stabilizers, non-stabilizers, or destabilizers, which may reflect different binding modes. To obtain more information about the mechanism of action of these fragments, we developed a competition assay using MST trick with the kinase tracer T236. The kinase tracer excitation emission wavelengths are compatible with the Dianthus device, and have been described as binding to the ATP binding pocket of different kinases, including PIM1 and PIM2. Therefore, we first determined the PIM3 affinity for the tracer, which was around 1 micromolar, and we fixed the final PIM3 concentration at 2 micromolar, sufficiently high for a complex formation. And then competition for the ATP binding site was tested with a selection of 6 fragments and our positive control AZD1208. 3 fragments were identified as potential allosteric binders. They did not induce changes to the MST trick signal, and 3 fragments were identified as ATP competitors. It is interesting to note that many kinase tracers are commercially available for different kinds of kinase, allowing the identification of ATP binder for a large variety of kinases. To complete this study, ADP-GLOW kinase assays are being performed to evaluate the effect of PIM3 binders on PIM3 kinase activity. In addition, crystallization attempts are currently ongoing to obtain additional atomic level information of the binding of the different fragments, which is the starting point for the fragment growing. That's it for the PIM3 fragment screening campaign. I hope it was clear, and I'll let you, Vanessa, introduce the second topic of this webinar, entitled The Design of PIM3 PROTAC.
Thank you, Maud, for presenting the ongoing PIM3 fragment screening. So let's move to the third strategy, that is the proof of concept of PIM3 PROTAC. PROTAC for proteolysis targeting chimeras were reported for the first time more than 20 years ago. They are hetero-bifunctional molecules that contain 3 components: the protein of interest, POI, a linker, and a E3 ubiquitin ligase binding moiety. This molecule can simultaneously bind the ligase and the target protein. Its ability to form this ternary complex is indicated by the cooperativity factor that corresponds to the ratio of a PROTAC's binary to ternary KD values. This factor generally gives a good indication of how favorable or unfavorable the complexes are. If you take a look on the picture, in cells, this ternary complex formation is the first key mechanistic step that results in target ubiquitinations and lead to its degradation via the ubiquitin proteasome pathway. Here on the slide, you will find all of the global Eurofins discovery capabilities regarding PROTAC characterization, from PROTAC permeability to cell-based assays. So now let's focus on the ternary complex characterization. We use the well-described PROTAC MZ1 and AT1 as an example to show you how the ternary complex induce drastic change of affinity for the protein of interest. As depicted on the left side, MZ1 or AT1 bind BRD4 BD1 domain and the ligase VHL. Binary interaction were determined either by SPR and spectral shift and show both very similar affinity in nanomolar range affinities. Regarding spectral shift data, the ternary complex was also formed, and you can see clearly a shift from the purple dose response curve to the blue one, adding the VHL as additional partner in the assay. At this range of affinity, we couldn't determine accurately the affinity. However, we could easily assess that the cooperativity factor is higher than 20 for AT1 and MZ1. In other words, the PROTAC bind 20 times tighter BRD4 in presence of the ligase. So now we will move forward to our topic, that is to generate PIM3 PROTAC. I will let Mo to describe you the strategy and present the data obtained for the ternary complex formation.
Thank you, Vanessa. Thanks to the X-ray structure of AZD1208 analog in complex with PIM1 that has been published, our chemists identified a potential exit vector for the design of PIM3 PROTACs. The kinase inhibitor was then derivatized with a pomalidomide linker moiety, leading to the PROTAC 154. The second PROTAC, named 136, was also synthesized with a different linker. A model of PIM3 CRBN E3 ligase complex using the AlphaFold prediction of PIM3 structure was generated by our chemists, and we performed the characterization of PIM3 PROTAC and CRBN PROTAC binding using biophysical approach to validate the model. First, we check the binary interaction between PIM3 and the PROTACs by TSA. As previously shown, the reference compound AZD1208 stabilized PIM3 by more than 18 degrees. Interestingly, the two PROTACs generated exhibit also significant stabilization of PIM3, more than 10 degrees, showing favorable binding energy despite their bigger size. To further characterize the binding of PROTACs to PIM3, we performed the binary interaction studies using spectral shift technology in order to obtain quantitative information. The setup was the same as the one used for the fragment screen. PIM3 was labeled with the histag labeling kit Redtris NTA, and the dose response of the PROTAC were tested with a twofold dilution factor. At the top of the chart, as you can see the result of our positive control, ATP gamma S, which gives, as expected, an affinity in the micromolar range. And then regarding the PROTAC results, as you can see, both PROTAC are high affinity binders. In fact, we reached the limit for an accurate determination of binding affinities because the measured affinities are higher than the concentration of PIM3. Nevertheless, this experiment confirmed the high affinity of the PROTACs to PIM3. We also characterized the binding of the two PIM3 PROTACs to the E3 ligase CRBN using spectral shift technology. CRBN was labeled with the labeling kit NHS thread, and dose response of PROTACs were tested with a twofold dilution factor. As you can see, the result for the pomalidomide moiety of the two PROTACs bind to the CRBN with an affinity in the dozen of nanomolar, which is in agreement with the literature. Finally, we characterize the formation of ternary complex using the following setup. We use the labeled CRBN at fixed concentration, 5 nanomolar, minimizing the protein consumption and enabling an accurate determination of binding affinities of the ternary complex, which are in the dozen of nanomolar. We could then determine the cooperativity factors, respectively 0.7 and 1 for PROTACs 154 and 136, meaning that there is no increase or decrease of the thermodynamic stability of the ternary complex. An optimization of PROTAC linkers and PIM3 warheads will be required to get a positive cooperativity. And now I hand it over to Vanessa to conclude and speak about the next generation of PIM3 PROTACs.
Thank you, Maud, for presenting the biophysics data for PIM3. It was nice to see the ternary complex formation, even if we would like to see even more cooperativity. To continue on this project, those PROTAC are also profiled in several tests, and particularly they will be tested in cells for the ability to degrade PIM3. After the validation of PIM3 PROTAC proof of concept, a second generation of PROTAC is ongoing with the new PIM3 binder moieties coming from the HTS. In fact, they were tested on the full kinase profiler platform, and some of those series show already good selectivity for PIM3. For example, on the right side of the slide, the hit displayed by the red dot bind only the PIM family. Other series do not show any selectivity and bind a lot of kinases as depicted on the picture on the left. As described previously, Biophysics will support the characterization of the new generation of PIM3 PROTAC with the affinity for PIM3 binders, affinity for cereblon binders, and also the cooperativity of the ternary complex. To conclude this webinar, we have take-home messages that we would like to share with you regarding the role of biophysics in drug discovery. First, we are deeply involved in drug discovery programs, whatever if you would like to develop inhibitors or degraders. Biophysics assay bring valuable information in binding assay, mechanism of action, protein stability and thermodynamic, and binding cooperativity. Additionally, biophysics is used as a complementary approach with the kinase profiler platform, which lists up to 400 kinase assays. For any question, request, or discussion regarding your project, feel free to reach us directly on this email address. I will hope to see you again for our next webinar focused on the membrane protein. The biophysics team is working closely with membrane protein experts from Eurofins Calixar to develop high-quality membrane proteins that are suitable for biophysics assays. It will be a very interesting and challenging topic to discuss with you. In the meantime, we wish you all a happy holiday season and a great start for next year.
Thank you, Vanessa and Maud, for your excellent presentation. We now have the pleasure to introduce our question and answer session where today's speakers will answer all of your questions. Don't forget you can still submit your questions using the questions panel in the menu on the right. So our first question is, how long did it take to perform the biophysical cascade?
for feasibility, from the feasibility to the mode of action. From the
Maud, we can't hear you at the moment. If you could please try again with your audio. There seems to be a little bit of feedback there.
Can you hear me now?
Yes, we can hear you now. Yep. And just move your camera because your camera's just tipped down there.
Okay. Yeah, I will switch it off because I have
some technical issues.
Sorry.
Okay, thank you.
For the feasibility to the mode of action, it took less than 2 months.
And the second question, do you recommend SPR or spectral shift for identification of ATP competitors for kinases?
That's a good question. Actually, we recommend to use both, but we have a preference for spectral shift data as there are a lot of tracer that can be used for a lot of kinase, and we can perform spectral shift easier. It will be easier to perform this type of assay with spectral shift. With SPR, we are consuming more ATP, and we have to add ATP in the running buffer to fully saturate the binding site. So I will recommend you to use spectral shift.
Did you check the effect of the PIM3 PROTACs on PIM3's degradation?
No, not yet. It is ongoing. We are testing the effect of the PROTACs on PIM3 activity, and our chemists are also developing new PROTACs. So we will have 30 PROTACs, new PROTACs to test.
How do you interpret the destabilization effect of the fragments on PIM3?
So it is now well admitted that destabilization, like as a stabilization, is due to the binding of the fragment, and it can be due to the binding to the less stable conformation of PIM3.
Could we apply fragment-based screening strategy to other families of proteins?
Yeah, so we can use fragment screening for other type of proteins like helicase, membrane protein, IDP, transcription factors. So yeah, it's quite easy to perform a fragment screen, and actually we demonstrate that with spectral shifts it's quite easy to perform this type of screening. And our next step now is to develop off-the-shelf helicase assay to accelerate the drug discovery programs, and to have some assay already off the shelf to perform fragment screening.
And this question is for Vanessa. Could you provide more details or clarify your question about the negative graphs related to the SPR analysis of PIM3?
Yeah, that's this question came often, I will say. It's weird, but if we take a look on the data and how SPR works, the negative data means something physiologically, and our interpretation about those data is the protein have some structural shape, and when it's immobilized on the chip and there is a binding that occur, we can observe some modification, some structural modification of the protein, and that leads to some change in the refractive index, and that's why we observe some negative signals So yeah, it just here in our case, PIM3 is not working because probably the binding, when there is a binding of ATP upon PIM3, there are some structural change. So by SPR, we are not able to see it. And there is no this issue with MST or spectral shift as the signal is linked to a modification of the fluorophore. And that's why here we are using the dianthus for screening.
This question is for Maud. Any data of ternary complex formation on SPR systems? Is there any data of ternary complex formation on SPR systems?
So for the PIM3, no, not yet. We are working on that because we cannot immobilize PIM3. It will require much more protein and to inject, and so and a new setup development. So we are working on that. It's something.
And for the PIM3 PROTACs coming from non-selective series based on the kinase profiler platform, are you planning to check if the degradation is more selective than the binding to the kinases?
It's not planned for now. It's a good question. But I will say first we will do the degradation assay, and then depending on the data that we will obtain, we will take a look also on the other the binding with the other kinases.
This question is for Maud. What CRBN construct were you using for the cooperativity studies? Was this the full-length CRBN/DDB1 complex, the ligand binding domain only, or another construct?
So it was the heterodimer, yes, CRBN DDB1 complex that we use, which was active, yes.
And how are you sure that the phosphorothioate ATP is a good analogue for ATP?
Yeah.
I will say that our standard when we are using SPR or biophysics in general, we like to work with no hydrolyzable ATP. There are other types of ATP used as a control. Here it was working pretty well with this ATP gamma. Do you have something else to add? So yeah, it's to avoid the autophosphorylation.
As there are no further questions, please join me in thanking Vanessa and Maud. As you leave the webinar today, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.